This is screen capture video unit 11.4. This is the fourth video in the series, and in the last video I wasn't watching the time and I ran out, but I was talking about it being an interesting time historically and also sociologically and anthropologically. I should mention, I believe it was National Geographic a number of years ago did an article on eye color and physical traits of some of the people in the stand countries in Asia, thinking that some of the people that are seen there today because of their eye color and skin color, mostly eye color, are descendants of Marco Polo and or his men as they traveled to and from China. But that would be for another topic and just interesting to mention. It is also interesting to note that environment affects the expression of the alleles, or the effect of the phenotype, because the phenotype is the expression of the genes. From the text, here are two females. I think they're from Middle Eastern, Iranian origin, and they're supposed to be identical twins, but some of you probably know environmental factors will affect identical twins, so that identical twins don't always look exactly alike. You can see that these two ladies are very similar in their features, but their differences, some of it is by the way they wear their hair and their clothing, but even so, there are some slight differences based on environment. Could be nutrition before birth, could be one had a richer blood supply than the other, could be nutrition after birth. It could be a number of things. But interestingly, identical twins are nature's clones. They are identical in their genetic makeup. This illustration shows what can happen in meiosis by independent assortment of the chromosomes and the alleles that they contain. And in metaphase 1, the chromosomes line up along the equator of the cell, and then in anaphase 1, they separate. Well, it just depends on how they line up and separate as to what goes into the daughter cells and the eventual gametes. What they're trying to show here are the ratios that will occur. 1/4 will be big R, big Y, 1/4 little r, little y, 1/4 little r, big Y, 1/4 big R, little y. And then eventually, when you self-fertilize these, the organism that contains these gametes, you're going to get a 9:3:3 phenotypic ratio. So that 9 of the 16 will be round and yellow, 3 of the 16 will be round and green, 3 of the 16 will be yellow and wrinkled, and 1 out of the 16 will be green and wrinkled. Linked genes add a new dimension to heredity, because so far we've thought about alleles on different chromosomes separating independently. But if the genes are linked on the same chromosome, they don't separate, they stay together. And that's what this illustration is trying to show. In this illustration from the text, we can see an explanation of linked genes. There's the purple flower color and long pollen, two genes that are found on the same chromosome, and red color and short pollen found on another chromosome. No, these are homologous chromosomes, so they have the same characters coded for on each one. There's one for color of the flower and the length of the pollen. But it just so happens that those two characters are on the same chromosome, so they don't sort independently. The only gametes that each can produce would be big P, big L, and little p, little l. And what you get then would be this 3-to-1 ratio. 3 out of 4 offspring are going to be purple flowered and long pollen. 1 of the 4, or 25%, will be red flower color, and short pollen. And what is not accounted for here would be purple round and red long. This seems to be missing, and the reason being is because these are linked genes. And so you only have this 3-to-1 ratio instead of the 9-to-3 to 3-to-1 ratio. And you could get some purple round and red long if there's crossing over. And we talked in the Unit 10 about tetrads forming and crossing over at the chiasma. And that's what they're showing right here. And if the crossover event occurs between the two genes, then you get recombinants form. That's what they're called, recombinants, that look different than the parental types. So the parental type would be big A, big B, and little a, little b. The recombinants would be a big A, little b, or little a, big B. And you can see with the color of the chromosomes how they're called recombinants. They do look different than the parental types. And those are the gametes that can be formed. Well, a lot of studies were done about 100 years ago with Drosophila flies, fruit flies, the common name. And they looked at such characteristics as their size, whether male or female, the wing length, whether they're hairy, whether they were light or dark, the length of these antennae, as you might call them, and eye color, just for a few characteristics. Now, just a hypothetical crossing over. If we have a female that's a wild type, she has a gray body and long wings, and then a male that would have a black body and vestigial wings. They don't function, crawl around, but doesn't fly. When you cross them, you might get some ratios such as this, showing close to 1,000 wild types, 1,000 of the black body vestigial wings, and then you get these recombinant phenotypes in much lower frequency. And what you could do is add the recombinant types and divide those by the parental phenotypes. And here they did it, 391 divided by 2300, and you get 0.17 or 17%. What that percentage means, what that number can be used for, is mapping out. This has been done a great deal with many organisms, whether it's fruit flies or corn or whatever it might be. They can tell a relative distance between the two genes. If they're close together, this number is going to be smaller. This percentage will be smaller because there's less chance that a crossover event will occur between two genes that are close together. If this is a high number, it signifies that the two genes are far apart. So here you can see where a crossover event might occur between the two genes to give these recombinants. Now here, the parental type, you're only going to get the one type of gamete. Sounds like my dog's really excited about this again. If we map these genes, just as I mentioned, it was found, these two genes, that there was a third gene signified with the letter C. And they used the 17% between these two FAR genes, and then they found that there was about 0.09 or 9% recombinant rate between the G and C, and about 9.5% or 0.095 rate between the C and the L gene. So that told them that the C was about halfway between these two genes. So those frequencies can be used in mapping. Now, partial gene map has been produced with the mutant phenotypes, wild type phenotypes. The short aristae, those are the antennae-like structures. Long versus black body, gray body. Cinnabar eyes versus red eyes. Vestigial wings versus normal wings. Brown eyes versus red eyes. The sex chromosomes become important when we talk about humans and higher forms of life that determine the sex. We understand that A male will have an X chromosome or Y chromosome. A female will have 2 X chromosomes. Some animals will have an extra X chromosome. Some humans have an extra X chromosome. Generally, for females, such as cats and mice and rats, only one of the X chromosomes will be expressed at one time, and that's one of the reasons why calico or tortoiseshell cats are all female. It's because at any given point in the cell, only one of the X chromosomes is being expressed, and the coat color for cats is on the X chromosome. So I was told years ago, you can count the number of color splotches on a cat. You can tell at what point during the development of the cat that it was producing those colors. It may have been at the 9-cell stage or 16-cell stage, or I should say 8 or 16, but supposedly you can count those and determine that. But all your calico cats are females. It's because of the 1X chromosome not being expressed at any given time during mitosis events. Well, here is an illustration, male versus the female, 44 chromosomes plus XY, female 44 chromosomes and 2 X's. And the male can give 22 chromosomes and an X, or 22 chromosomes and a Y in each of the sex cells. Female, 22 chromosomes and an X, one of those X's. And then the offspring are either going to be female or male, depending on whether they have 2 X's or an X and a Y. In other organisms, such as crickets, roaches, grasshoppers, they have a different system for determining the sex of the individual. It's an exosystem, where a female would have 2 X chromosomes and the male would just have an X, but no other chromosome. That's why it's called the exosystem. There's also a ZW system, and we see this in some birds such as chickens and some other animals, where the female has a ZW and the male has 2 Z's. Insects such as ants and bees, the sex is determined by chromosome number. So the double number, 32, would be females, and the males would just be the 16, the unfertilized egg. This leads us into sex-linked traits. Some traits are sex-linked, such as eye color in fruit flies. And so you see those show up with certain frequencies in males and females. Here a sex-linked trait is shown in the fruit flies. You have the female with the red type eyes, big R is red, and a white-eyed male with the little r being shown as white. You cross these two and you're going to get half females, half males. The male is going to get the normal gene from the female, so all the males will have red eye color and all the females will have red eye color. Now you cross those, the female, that's the heterozygous for the two, with a normal red-eyed male, and half the males are going to have white eyes, and half the females are going to be carriers. So XY be male, half of those males are white-eyed. Statistically, 50% of the males would be. All the females would have normal eye color. Now if you take a heterozygous female, she's carrying the white eye color gene, with a white-eyed male, and the only gene for eye color that he's carrying is for white. And you cross these, half the females will have white eyes and half the males will have white eyes. In humans, the royal family of Europe is an interesting study, and I'm just about running out of time, so I'll pick this up in the next video, and that should be my last video.